Welcome to this explainer. Today, we're diving into a microscopic transformation that is, literally, essential for a plant to secure its future. When we look out at a field of blooming flowers or a crop that's finally ready for harvest, we're really just witnessing the grand finale of a deeply complex developmental shift. So today, we're going to trace what you might call the biological hero's journey of a tiny, tiny cluster of stem cells. We're going to see exactly how it completely reprograms itself from endlessly churning out leaves to building a flower. But to understand this transition, we really have to start with one crucial question. How does a plant actually know when to make that switch? I mean, unlike animals, a plant can't exactly pack up and migrate when the weather turns bad or resources run low, right? Its reproductive success depends entirely on getting the timing absolutely perfect.
So, let's meet the protagonist of our story, the shoot apical meristem, or the SAM for short. You can think of the SAM as this microscopic stem cell reservoir. It's essentially the plant's built-in 3D printer. Located at the very apex or the growing tip of the shoot, it holds a population of stem cells that generate literally all of the plant's above-ground architecture. But its ultimate destiny, the real climax of its journey, is to transform into an inflorescence or floral meristem. For the plant to reproduce, this SAM has to undergo a radical, highly coordinated reprogramming.
To break this all down, we'll follow a 6-step agenda today. 1. The decision to flower. 2. Delivering the floral signal. 3. Hormonal and genetic reprogramming. 4. Reshaping the apical meristem. 5. Master regulators of form. And finally, 6. Birth of the flower.
All right, let's jump into section 1, the decision to flower.
Like we mentioned, getting this timing right is a matter of life and death. Take winter wheat versus spring wheat as a prime example. For winter crops, the plant actually needs exposure to long periods of cold, a process known as vernalization, along with an extended vegetative growth period. By overwintering and holding off on that decision to flower until the spring, these winter varieties can actually produce yields that are 50 to 100% higher than spring-sown varieties. So the plant is constantly calculating, checking if it has enough resources and if the environmental conditions are just right.
And to make that calculation, the plant relies on a truly massive sensory network. It is integrating an extraordinary amount of data. It tracks day length, that's photoperiodism, using photoreceptors like phytochromes for red light and cryptochromes for blue light. Then it aligns all that light data with its own internal 24-hour circadian clock through a master regulator gene called constans. Throw in temperature sensing and vernalization, and you've got 5 distinct interconnected pathways feeding data into the plant. They're all working together to tell it exactly when conditions are perfect to stop making leaves and start making the seeds.
Moving on to section 2, delivering the floral signal.
So, once that massive sensory network agrees it's go time, the plant has to send a message to our protagonist, the meristem. Now, the actual decision is made down in the leaves, where the light is perceived. Under the right long day conditions, this triggers the synthesis of a protein called FT. This is it, folks, the elusive florigen molecule that scientists literally spent decades searching for. This FT protein is physically transported through the plant's vascular system, traveling all the way up from the leaves to the shoot apex. And once it arrives at the meristem, it teams up with other proteins, like FD and 14-33. Together, this complex flips the master switches to begin reprogramming the stem cells.
Which brings us to section 3. Hormonal and genetic reprogramming.
What happens the moment that florigen signal arrives is a cascade of sheer biochemical complexity. It's basically a genetic power struggle, and it looks a bit different depending on the species. In soybeans, for instance, we see a huge shift in hormonal pathways. Abscisic acid, auxin, jasmonic acid, all cross-regulating the meristem's activity. In maize, you've got specific regulatory modules and genes like unbranched fine-tuning the transition. And then in the model plant Arabidopsis, master transcription factors like SOC1, AP1, and LFY get rapidly upregulated by florigen, while repressor genes like SVP are actively dialed down. It is a massive, highly dynamic change in gene expression that completely rewrites the meristem's identity.
And that triggers section 4, reshaping the apical meristem.
Because once the genetics are reprogrammed, the actual physical scaffolding of this biological construction zone has to change. This is the real plot twist of our cell's journey. During the vegetative phase, the meristem is pretty flat, and it exclusively pumps out leaves. But as the floral transition takes over, boom, there's a huge surge in mitotic activity. Cell division hits the gas pedal. The meristem increases its cell population, its distinct central and peripheral zones start to enlarge, and it physically mounds up. It goes from a flat leaf-making factory into a rapidly dividing domed structure that is ready to build a flower. When we actually map out these 3D morphological changes in the Arabidopsis meristem, you can literally watch this physical doming and volume expansion take place. By using fluorescent domain mapping, we can see exactly how the different genetic zones of the stem cell niche physically balloon outward to accommodate the complex architecture that a flower requires.
Let's look closer at how that works in Section 5, Master Regulators of Form. You might be wondering, how does the plant control this shifting, expanding architecture without just losing its stem cell reservoir completely? Well, it maintains the scaffolding using a brilliantly delicate negative feedback loop between two key domains. Down at the base, you have the organizing center. This produces a transcription factor called WUSCHL, or WUS. WUS sends an up signal into the central zone right above it, basically telling those cells, hey, stay as stem cells. But to prevent runaway growth, those stem cells produce a peptide called CLAVATA3, or CLV3, which sends a down signal to restrict that WUS expression. It is a perfectly balanced loop. It ensures the meristem grows to the exact right size without spiraling out of control.
If we track this on a timeline, we can see exactly how the plant choreographs this shape change. Around day 10, that central zone begins to expand. By day 13, the organizing center's height transiently peaks, which pushes the whole meristem into that dome shape. Now, a transcription factor called AP2 is actually the mastermind behind this rapid growth. But here's the clever part. By day 16, to finalize the inflorescent shape and stop the meristem from overexpanding, AP2 is actively repressed by SOC1. Remember SOC1? That's one of those master floral genes we talked about. This reciprocal repression acts as an elegant biological braking system, perfectly synchronizing the meristem's expansion with the onset of flowering.
Which brings us to the grand finale, Section 6, Birth of the Flower. The hero's journey concludes with the birth of a totally new organ. The meristem has now achieved what biologists call a determinate identity. That means it finally stops its continuous, indefinite growth. Having successfully morphed its internal blueprints away from endless vegetative leaf production, it now executes a finite reproductive architecture. It initiates an exact, strictly defined number of sterile organs, those are your sepals and petals, and then the vital reproductive organs, the stamens and carpels. The transformation from shoot apical meristem to floral meristem is entirely complete.
As our source material beautifully summarizes, the structure and function of the SAM change with plant age and in response to environmental signals, causing it to form flowers and inflorescence. This highlights the truly elegant reality that all above-ground plant life, and honestly, the agricultural systems that we all rely on to survive, are shaped by this highly coordinated microscopic dance. It is a stunning integration of external environmental signals and internal genetic scaffolding.
As we wrap up this explainer, I want to leave you with a thought to ponder. We've just seen how unbelievably sensitive this transition is to precise temperatures, day lengths, and light quality. So as our global climates rapidly shift, understanding these delicate stem cell pathways isn't just fascinating biology anymore. It is absolutely crucial for the future resilience of our global food supply. How will these intricate pathways adapt? Thank you so much for joining me on this microscopic journey, and I hope this provided a clear, engaging look into the wild and incredible world of plant development.